much, Dr. Levy, for the kind introduction, and thank you to the IBSA Foundation for having me today. Dr. Bianco sends his best to you all, but we'll go ahead and get started with some of the research from our lab. So the objectives of our talk today are to characterize the phenotype of a mouse with a specific inactivation of the type 2 deiodinase within its hepatocytes, and to define the metabolic impact of D2-mediated T3 production in the liver throughout development and adulthood. I have no disclosures. So the thyroid gland produces and secretes thyroid hormone in form of both T4 and T3, where T4 is the prohormone, but T3 is the active form of the thyroid hormone. These circulate in the periphery, and what's important to remember about the concentrations of T4 and T3 in the circulation, in particular, is that the levels of T3 in our blood are very stable throughout life. Except in the settings of severe illness or fasting, T3 levels are very stable. But in the cell, we know that it's a rapidly changing environment with increasing and decreasing metabolic demands. So the fact that the T3 levels in the circulation are stable suggests that peripheral systemic levels of T3 are not directly responsible for those rapidly fluctuating metabolic changes within the cell. So the key to thyroid hormone metabolic control in the cell is the deiodinases. So you can see that T4 and T3 enter the cell, as Dr. Visser explained, where they can encounter the deiodinases, D1, D2, and D3. So once the thyroid hormone is in the cell, and it may or may not encounter one of the deiodinases, the T3 action can occur. And today we're going to be discussing one very important T3 target organ, which is the liver. The liver is a classic example of a T3-dependent tissue. We know that T3 in the liver stimulates lipogenesis, fatty acid oxidation, bile acid synthesis, cholesterol metabolism. We know that from clinical studies in cases of thyrotoxicosis that T3 induces hepatic glucose production and decreases hepatic insulin sensitivity. And we also know that the liver is an important site of thyroid hormone clearance through both conjugation and through activation of the D1 pathway. So because the liver is an important T3 target organ, this has led many investigators to wonder whether there is a role for the type 2 deiodinase within the liver. This is the deiodinase that activates T4 and turns it into T3, which can then bind to the receptor and have impact. So what we know from a large range of studies is that there is D2 expressed in liver of certain animals. There's D2 in the liver of chickens and amphibians during early stages of metamorphosis. There's D2 in the macrophages that live within the liver of mice. And there's ectopic D2 found in mice with knockout of the liver X receptor. But D2 is not known to be expressed normally in the adult mammalian liver. So we have a global D2 knockout, so it's a D2 knockout mouse that has the inactivation of the D2 in all of its tissues. And when we take wild-type mice and we put them on a high-fat diet, you can see that the liver tissue doesn't contain much fat, which is shown in red oil O. Those are those red droplets there. There's not much fat in the Chow diet liver of the wild-type mice. But when you place them on a high-fat diet, of course, steatosis is induced and you get those nice red droplets. But when you look at the global D2 knockout mouse, you see something very interesting. That, first of all, at baseline on the Chow diet, there is more fat in the liver of those mice. And then when you place them on a high-fat diet, there is even more fat. So the global D2 knockout mouse has liver steatosis at baseline, and this is induced even further by placing on a high-fat diet. But how could this be if there's no D2 in the liver? What is responsible for this phenotype? So it was explained that this could just be secondary to other peripheral effects from the D2 knockout via issues with the knockout in the brain or the brown fat or skeletal muscle, other metabolically relevant tissues that somehow is acting on the liver secondarily. So to study this further, to see if there's really a role for D2 within the liver itself, we needed to create a mouse that specifically was lacking in D2 within the hepatocytes. So we did this by crossing the FLOX-D2 mouse with the albumin-Cree mouse and created a mouse that we're going to refer to throughout the talk as the albumin-D2 knockout mouse. In other words, specifically lacking D2 within the hepatocytes. So what is the phenotype of this mouse? So you can see that both at P1, which is postnatal day 1, the baby mouse, and an adult mice, that control and albumin D2 knockout mice are systemically euthyroid, have the same levels of T4 and T3 in the periphery. The males and females have normal growth and development compared to controls. Their body fat composition and bone density is the same as controls. And their livers weigh the same amount, have the same gross morphologic appearance, and the same histology. So at baseline, there isn't much of a phenotype that we see. So we wanted to see if we could provoke a phenotype. So we placed these mice on a high-fat diet and found something interesting. So when you take the control mice on Chow diet, you can measure their body weight. It increases a little bit. But when you put the control mice on a high-fat diet, their weight goes up dramatically. They get obese. When you do the same with the albumin D2 knockout mice, you see that those on a high-fat diet don't gain any extra weight. In fact, they're protected from diet-induced obesity. So we wanted to make sure that we really believed that and kind of measure this in different ways. So if you look at the epididymal white fat deposit, you can weigh the tissue there, and you see that in the control mice, the level of white fat in the epididymal area goes up significantly on a high-fat diet, but not so much in the albumin D2 knockout mouse. The liver weight goes up dramatically on a high-fat diet in the control animals, but doesn't increase at all in the albumin D2 knockout animals. And you can see that placing on the high-fat diet for the control mice results in a significant increase in the proportion of body fat in the control animals, but this, again, does not occur in the albumin D2 knockout animals. If you look at the blood serum cholesterol levels, lipid levels in these animals, you see that when you place the controls on the high-fat diet, serum cholesterol levels increase, but to a lesser degree in the albumin D2 knockout animals. But triglycerides is quite interesting. Triglycerides go up on the high-fat diet in the control animals, but don't increase at all in the albumin D2 knockout animals. So they also seem to be protected from hypertriglyceridemia. So if you want to look at the histology of these mice, you see that in the control animals, the Cree albumin animals, on the Chow diet, there aren't any clear, obvious lipid droplets within the hepatocytes. But when you place on the high-fat diet, which we just said causes obesity and hypertriglyceridemia, you see a nice histologic structure there as well with those clear lipid droplets very visible in the cells. But consistent with our other findings, these do not happen even in the high-fat diet in the albumin D2 knockout cells. You can actually measure the lipid levels within the liver tissue itself, and you see that cholesterol levels within the liver increase dramatically in the control animals, but to a lesser degree in the albumin D2 knockout animals. And the liver triglycerides show a similar story, a greatly increased induction in the high-fat diet and the controls, and to a lesser degree in the albumin D2 knockouts. So a brief summary of what we've shown so far from the phenotype of these animals is that a liver-specific D2 inactivation prevents diet-induced liver steatosis, obesity, and hypertriglyceridemia. But how is this possible if D2 isn't even expressed in the adult mouse liver? So this led us to a new hypothesis that perhaps loss of D2-dependent processes during liver development induce this phenotype that sticks around into adulthood. So we started studying mice at different embryologic developmental days, and we were able to detect D2 mRNA with quantitative PCR by about embryologic day 12. Those levels increased to a peak at postnatal day 1, P1, to about threefold compared to that of E12. And they rapidly declined thereafter in a few days to reach the levels of the adult, which are undetectable. So you might say, well, why didn't you just stain the liver for D2? But the problem is that we don't have a good antibody for D2 immunohistochemistry. So we proceeded with in situ hybridization of this liver tissue. So if you take this liver tissue from P1 mice, which is when there's the peak in mRNA levels, you can see here the H&E staining of the hepatocytes. And in C and D, you can see those white blotches that look like stars. That is the D2 showing there. And you can see in P1 liver, it looks like you're looking into the Milky Way. There's a lot of stars there. The D2 is diffusely expressed throughout the liver. But if you then take adult liver, which does not have detectable D2 mRNA, and you do in situ hybridization, you can see that the sky has gone dark. So what are the deiodinases doing in the control liver? So when we're talking about D2 and the deiodinases in general, we don't like to just focus on mRNA, because something that we really care about is the activity of the enzyme. So even though we detected mRNA in these P1 mice, is there activity of the enzyme? So if you take the liver from the P1 mice, and here we're just looking at wild type and control, the Cree albumin animals, there is detectable liver D2 activity in P1 mice. There's also D3 and D1 mRNA. D3 levels fluctuate throughout embryologic development, whereas D1 levels increase steadily to reach very high levels in the adult, which we know. But now, if you start looking at the D2 activity in the albumin D2 knockout, you can see that it's about half that of the controls, maybe slightly less. But there is some detectable D2 activity still in these animals, especially compared to the global D2 knockout, which has 0 detectable D2 activity. And what this should mean to you is that in P1 liver, there is some other cell that's not the hepatocyte that has a little bit of D2 activity. The D3 and D1 levels mRNA were the same in the albumin D2 knockout mice as the controls. And if you look at the D2 mRNA levels, you can see that this peak at P1 that we observe in the controls is decreased by about half in the albumin D2 knockout animals. So how is this affecting T3 levels? So you can actually quantitate the amount of T3 produced in this P1 liver, and you see that albumin D2 knockout mouse do have significantly less T3 production than their controls, whereas by postnatal day 5, they have the same amount of T3 production. And you can measure liver T3 content where the T3 is less in albumin D2 knockout animals. So this decrease in D2 levels and activity in P1 does translate to a difference, a decrease in T3 at those time periods as well. So we wanted to know what this is doing to the liver. So we chose some candidate genes that we know are T3 sensitive genes or genes involved in lipid homeostasis, and we measured the mRNA at different time periods throughout development. And in the control mice, we found that on many of them, the pattern was consistent, meaning that they increased from E12 up to P1 and then even higher into adulthood. But what was really amazing about these candidate genes that we had chosen is their expression pattern in the albumin D2 knockouts was very similar. And what we found that was in each of them in P1, their expression was much less. But the difference completely dissipates by the time the mice are adults. But the phenotype that we're seeing is in adult mice. So how is this change in gene expression during P1 affecting the adults, which did not show a difference in these genes. So we wanted to then move towards a rather unbiased whole genome approach and go ahead and perform microarray analysis of the adult livers. And what we found was interesting. Here you can see a heat map of the albumin D2 knockout mice and the control livers, and you can see there's a clear difference in gene expression pattern. And this, again, these are the adult mice. We found 165 differentially expressed genes in the albumin D2 knockout mice that were significantly differentially expressed. And what we like to do when we're looking at microarray analysis is not only to consider those 165 genes separately, but to try to understand what kind of physiologic relevance they have together and try to put them under umbrellas so that we understand their relevance. So we use something called gene set enrichment analysis. So the gene sets that were enriched in this analysis were related to lipoprotein binding, carbohydrate transport, inflammation, and methyltransferase sulfurtransferase. So we have updated our hypothesis based on these results. The presence or absence of perinatal D2 generated T3 in hepatocytes permanently modifies gene expression and defines a phenotype that is carried forward throughout adulthood. So we wanted to know if there is some sort of epigenetic situation happening here. So we went ahead and performed whole genome DNA methylation profiling. So again, we took the livers from adult albumin D2 knockout mice and controls, the Cree albumin mice, and sent them to our collaborators for this methylation profiling. And remarkably, they did find more than 3,500 differentially methylated regions, or DMRs, within more than 3,000 genes that were different. You can take these DMRs that we found that were different, the 3,500 DMRs, and you can actually map them to different chromatin states. So the question is whether any of the DMRs that we found that were significantly differentially expressed were on either active or repressed chromatin sites. And about 20% are on active or repressed chromatin sites. And you want to know what is the relevance of those DMRs. And similar to the gene set enrichment analysis that you can do with microarray, you can do great analysis to try to understand the physiologic importance of these specific DMRs that are different in your samples. And we saw that the DMRs that were located on the active chromatin sites were related to abnormal hepatobiliary physiology, abnormal liver physiology, hepatitis, decreased HDL levels. And those DMRs that were located on repressed chromatin sites were related to embryonic morphogenesis, embryonic organ development. So those studies that we just discussed about the transcriptome and the methylome of the mice of adult albumin D2 knockout livers were taken from studies of those animals on a Chow diet. So now we wanted to look and see if there was anything different in those on a high-fat diet. So again, we chose candidate genes that are T3 sensitive or involved in lipid homeostasis, lipid metabolism, and we wanted to look at how those changed when the animals were placed on a high-fat diet. So here you can see one gene in particular that is induced by a high-fat diet in the control animal. And you see that its induction is impaired in the albumin D2 knockout animal. And this pattern was consistent for many of these genes. There seems to be just an impaired induction of many of the genes when the animals are on a high-fat diet. We also performed, again, the microarray analysis and the DNA methylation profiling of the animals on a high-fat diet. And again, you see that there's a nice heat map here between gene expression patterns in those animals that are the albumin D2 knockout on a high-fat diet versus those controls on a high-fat diet. And we found, again, about 150 genes that were differentially expressed. And what's amazing is that most of those genes are completely different than those found in the animals on a Chow diet. So there's different gene expression patterns depending on the animal's diet. we then wondered whether the DMRs, the 3,500 DMRs that we found in these animals, were any of those regions on the regions that, of the genes that were affected in the microarray. So we did a nice little Venn diagram here and see that about 10% of the differentially expressed genes contained DMRs. So in conclusion, D2 is briefly expressed in neonatal mouse hepatocytes, and disruption of this D2-mediated T3 signaling protects adult mice from diet-induced obesity, steatosis, and hypertriglyceridemia, reduces the expression of genes in the neonatal liver involved in fatty acid, triglyceride, and cholesterol synthesis, and permanently modifies the liver transcriptome via epigenetic mechanisms that involve DNA methylation. So I'd like to thank my mentor, Dr. Bianco, who unfortunately was not able to be here today, but allowed me to have this fabulous trip to Naples. My lab members, Dr. Tatiana Fonseca, who's the first author on this paper, Sungro, Danielle, and our wonderful collaborators, Dr. Garabin and Dr. Wang. Thanks so much.